Большое спасибо. Good morning, everyone. Guys, start coming to take a look at the lecture in microbiology. And что, не слышно? Или посильнее?
Все нормально. Просто подумал, что в соседней комнате тоже вроде третья аудитория. Надо проверить.
Не, у меня вторая.
Вторая у вас?
Я же сказала, у меня вторая.
Все, я прошу прощения.
Да-да-да, посмотрите, да. Я тоже. У меня там было зачеркнуто, но вторая написана. Good morning, everyone. We go on with our lectures in special microbiology. Guys, start coming for a lecture. If you would like me to inform you through VK what room and what topic is today, or I can see that some students are coming up to hear the lecture. Good morning, my dears. So today we go on with the special microbiology. And you've heard several lectures on this part of microbiology course last year. You heard lectures already about fungi, and also you heard lectures about the Staphylococcus and Streptococcus gram-positive cocci, and about Escherichia coli, gram-negative rods, plus Salmonella and Shigella. So now we start with the microbes that may be a real threat for human being. These are bacteria that are recognized as possible warfare agents. So it means these organisms have a very strong potential to be used as biological weapon. Of course, I hope in your doctor's practicing, in your career, so you would never see these bacteria or diseases caused by these bacteria. Maybe except for Vibrio cholera, because cholera still is endemic in India and some African countries. But then as to Yersinia pestis that causes plague and Bacillus anthracis that causes anthrax, I hope even if we take into consideration that there is some morbidity due to those diseases, so you would never see them in your medical practicing. But still, because they can be used as warfare agents, you should know at least, we should cover at least some general important information about these three types of microorganisms. So today we will talk about Yersinia pestis and plague, Vibrio cholera, and the disease caused by this microbe that is called cholera, and Bacillus anthracis, that is a causative agent of anthrax. But before we start discussing particular microorganisms, I would like to introduce you to the general concept of biological hazard or biohazard. And this is a biological substance that poses a threat to the health of living organisms, mainly and usually humans. But those substances also can be harmful for animals. This could include a sample of microorganism, virus, or toxin that can adversely affect human health. So, as an example, microorganisms, bacteria, these are the Yersinia or Bacillus. Viruses, these are coronaviruses or Ebola viruses or variola viruses, or toxins, and mainly we consider the toxin of Clostridium botulinum. Botulinum toxin is one of the most potent poison thing in the world. We won't talk today about viruses or toxins. We will talk today about bacteria only. And then at the end of our course, we'll discuss viruses that can be potentially used as a warfare agents. Okay, now let's take a look at this diagram. And usually, to describe the precautions, safety precautions that are necessary to be applied in the microbiology lab, depending on the specific pathogens handled when performing laboratory procedure. So this is the appearance of the iceberg, right? And according to USA classification, United States of America classification, and Western world, the highest risk microbes are placed on biosafety level 4, and the lowest risk microbes are placed on biosafety level 1. Interestingly, that, and you have to know this while you study in the Russian Federation. Then in the Russian Federation, we have everything upside down. So, for example, our laboratory in our microbiology department is assigned for biosafety level 4 and 3. And according to Russian classification, so the lower risk microbes are placed in biosafety level 4 and 3. And highest risk microbes are placed on the level biosafety level 1 and 2. But because you studied in English, but you have to consider it, you have to know this. But because you study in the in English, so we would stay with this diagram of biosafety levels. And let's look at the microorganisms that are placed on those biosafety levels. The lowest risk will be seen when we are working in the laboratory with the non-pathogenic strains of E. coli. Non-pathogenic strains, because you have to know already, because you had lecture about Escherichia, that we may have highly pathogenic Escherichia coli with oosomatic antigen or 157. It is placed on biosafety level 2. But non-pathogenic Escherichia coli belong to biosafety level 1. Then on the biosafety level 2, we have Shigella, Salmonella, the causative agents of typhoid and paratyphoid fever, Staphylococcus aureus, Streptococcus pyogenes, quite a lot of bacteria that you have seen in our department. So because we can work with this, we are allowed a license to work with this group of microorganisms that under some conditions can cause quite severe diseases. Then we go up for biosafety level 3. Mainly all bacteria that cause serious diseases are placed on this level. For example, Mycobacterium tuberculosis, then Vibrio cholera and Bacillus anthracis. They are placed on this biosafety level 3, while the Yersinia pestis is the only bacteria that assigned for biosafety level 4, because it is a microbe with a very high risk of causing very serious lethal infection. So the complexity of each level aligns with the infectivity, disease severity, and the microorganism's ability for transmission, including exposure routes, and the nature of the laboratory work to be performed. And also, what is not written here, the important features of the microorganism that is taken into consideration when we place it into a particular level is whether the treatment is developed or not very well and whether the treatment is effective. Then the second, whether we have specific prevention measures like vaccination against this infection or not, and for how long the vaccination may give protection. And that is also important. And one more thing, how easily this microbe can be genetically modified. Because I would like to give you example with Yersinia pestis. Truly, now with the development of many groups of antibiotics, uh, plague as a disease can be treated successfully if we start treatment in not without very serious delay. But then the problem is that if someone is going to use it as warfare agents, Yersinia pestis can be genetically modified, and some genes can be cloned into Yersinia genotype or genome that will give it antibiotic resistance. Then the good drugs that we usually use or we know can be used for the treatment will be not working properly. That is the problem. So this is also important feature of bacteria that or generally microorganisms that is considered when we find the particular level. So here you can see the table uh, related to the risk groups or to biosafety levels and practices and equipment. And here you can see the last column, this is safety equipment. What does it mean, safety equipment? So to contain the microproliferation, we have to perform some tricks and use some equipment, right? So, and the regular open bench in the laboratory can be used for biosafety, for working with the organisms from biosafety level 1 and 2, which are not highly pathogenic. So, and GMT is good microbiological techniques that you, by the way, tried to perform yourself already in the general part of microbiology. Then when we, and that's laboratory type is basic teaching or basic research. That's what we have. And also biosafety level 2 is related to primary health service, diagnostic service, research, and partially teaching. Then this is also good microbiology techniques plus that you use, that's flaming the loops and flaming the instruments and keeping controlling the contamination by flaming everything. That's what you know already. But also on the biosafety level 2, we need a protective clothing, biohazard sign, and as an equipment, open bench is usually enough. But if this microbe can be spread with the aerosol, then we need biosafety cabin for potential aerosols. When we go higher, for higher containment, biosafety level 4, so the usually students and pupils cannot enter that laboratory. So this is not for basic teaching or research. This is special diagnostic services. Uh, Usually we refer to them as reference laboratories or research. So laboratory practicing is biosafety level 2 plus the controlled access into the laboratory and directional airflow. You have to know that in the laboratory, the air should flow only in one direction from outside, and this because we have the a bit lower pressure in the laboratory, and the air flow is going from outside to inside and outside through the system of special filters, not to cause contamination and proliferation of microorganisms outside. So that is called directional airflow. And according to safety equipment, we should have we have to have the biosafety cabin of particular level and maybe other primary devices for all activities. When we go up to the highest maximum containment biosafety level 4, that's where really dangerous pathogens, we are dealing with really dangerous pathogens. And we need to add airlock entry, shower exit, special waste disposal with the air autoclave. So you can see here the double-ended autoclave. What is it double-ended? We load our autoclave from inside the laboratory. Then after the decontamination happens, the waste products will be released to outside the laboratory. That is very important to prevent contamination outside. So see through the wall and highly filtered air and other devices like positive pressure suits that should be or have to be weared in the laboratory. That's a general description, my dears. I think this is more than enough for general covering the biosafety levels. And now we come up to Yersinia pestis, the causative agent of plague, that belong to biosafety level 4 according to USA classification, or biosafety level 1 according to Russian Federation classification, because it makes a possible threat for all people who are working with it or exposed to it. So, generally saying, what is a plague? It is a serious bacterial infection that can be really deadly. Unless treated, a plague may have a mortality rate that may depend, or usually depend, on the type of the plague. Because according to the way of route of infection and the localization of the infection, we may develop several groups or several types of infections. Two of them are bubonic plague and pneumonic plague, which the second one is much more severe. And if not treated, it is 100% lethal. Bubonic plague, if not treated, 50% lethal. That's quite a lot anyway. So the natural source of this disease, so because this, we come up again to a question that some microbes may occur in nature, and we don't have any chance to eradicate these microbes from nature because they can infect animals, they can reside in the soil, in the rodent burrows. So they may reside in the vectors, which are the fleas. So then we have no chance to eradicate them. So the plague is always with us. Although the morbidity is very low, because we have several cases every year in the United States and Russian Federation, sometimes in Kazakhstan and some other republics in India, but still there's very few. But we may never get rid of it. So, because the natural source of this bacteria is the infection, the wild rodent that may carry this bacteria, and this bacteria may also cause infection in animals and outbreaks in animals that is called enzootic. Uh, So we have enzootic area around the world usually. So the vectors for this disease, for plague, is usually from animal to animal. It is transmitted by fleas, but also from animals to humans. It can be transmitted also by fleas, by flea bite. So let's take a look first. That was a disease. Let's take a look at the organism itself. Because we are studying microbiology, and the most important thing is characterization of the microbe itself and recognizing the microbe itself. So for a very long while, three microbes or three types of bacteria, Yersinia, Francisella, and Pastorella, had been placed in one large group because we have several common features in these species. First, they are animal bacteria, so they have animals as natural hosts, all these three. Then they can be transmitted from animals to humans and can cause diseases in humans. And according to the shape of this bacteria that you see here, they are all together pleomorphic gram-negative rods. So, and they exhibit so-called bipolar staining. So later on, we again take a look at it. But if you take a look here, and thank God you are sitting not in the lecture room, and there you could not see it, but if you take a closer look at your computer or cell phone, you would see that the bacterial resembles the safety pin that is darker at polars at the ends and lighter in the middle, right? Can you see? So this appears as safety pin. That is typical for all three, Yersinia, Francisella, and Pastorella. That's what we call bipolar staining. Then they have some common features in the fermentative tests. They are catalase positive. It means they may decompose the substrate, hydrogen peroxide, and oxidase negative. And they are also facultative anaerobes. Do you remember what does it mean? It means that they can live with or without access of the oxygen. Or some of them can be microaerophilic. So, but then out of these three, the most dangerous one is Yersinia as a genus, and the species Yersinia pestis, that is a causative agent of the plague. So we won't discuss Francisella and Pastorella anymore today, and we concentrate on Yersinia. So Yersinia caused the several very serious epidemics in the history of the mankind. The first very well described pandemic occurred in 6th century Anno Domini and killed 100 million people in 50 years. The second one, that is very well described in a lot of novels and poems, major pandemic that was called Black Death that originated in Asia in 14th century and spread to the Near East and Europe within 2 centuries. And it is believed that in Europe alone 1/4 of population died of the disease. And the third pandemic originated in Burma in the 1890s, spread to China, and from there to other continents. And the spreading occurs through rat-infested ships because the rats are as you can see here, so the rats make the natural source of the disease. So then, this spreading through rat-infested sheep, it led to the establishment of the organism in wild rodents in many countries along the world. So then I would like you to take a closer look at this painting, The Triumph of Death by Paul Bruegel, that was done in 16th century. And this picture shows the fear of this disease, and it shows also the inability to escape from this disease, even from those high levels. So, for example, you can see the king dying at the street. So that's usually the way how kings were shown by artists in their pictures. So then you can see the carriages of skulls, of human skulls, and you can see the hips over the human corpse, and you can see the hungry dogs eating those corpses on the streets, and the uh, mood or mood of this picture shows how the European citizens uh, mm, take the risk of to be infected with the plague. So then, how about plague today? You may think that it disappeared. No, it doesn't disappear. You can see here the countries and the summary of the infection that occurred within 2010, 2015. So you can see that it may occur in the United States, in the South America, in Bolivia and Peru, for example. Then somewhere in the African continent, in Uganda, Congo, Tanzania. So then on the in Mongolia, China, even some cases in Russia, Kyrgyzstan, and of course Madagascar is the very common place where it may occur from time to time. So then, as I said, from time to time, we have cases in many parts of the world. Uh, there are very few of them, 1, 2, 3 to 4 cases a year, but still they occur. And for example, one of the latest cases emerged in Mongolia and China in July 2020. 2 deaths occurred. And you can see here a boy who hunted marmots. These are animals that traditionally can be used for eating in Mongolia and China. And again and again, the government produced the prohibiting of the hunting these animals, but still the contact with these animals that can be natural source of the Yersinia pestis from time to time cause the infections. So in the Russian Federation from time to time also occurred some cases, and the last cases were due to the contact with the camels, and because the camels in particular areas of the world also can be the vectors for this disease. Now let's take a closer look of Yersinia pestis. So this is a small gram-negative rod, non-motile, non-sporing, non-acid fast. It means it has the typical gram-negative type of the cell wall. That's what we mean under bipolar staining. You see the ends are darker and the middle is lighter. And here you can see closer the so-called biosafety pin appearance here and there. So Yersinia pestis, my organism itself, is very sensitive to heat. So by boiling, we can kill it very easy. It is sensitive also for sunlight, drying, chemical disinfectants, and it can be easily uh, destroyed by 0.5% of phenol for 15 minutes. But usually it is resistant to cold and especially in moist environment. So that's where they may occur for very long while. So, and also it is believed that it remains viable for 1 year in the frozen infected fleas. So, and it remains also viable for several months in the soil or rodent burrows. That can be a source of a live Yersinia pestis, even if you would destroy all fleas and all rodents around. Still, bacteria may survive for a long while. So then now that your morphological characteristics, and now we describe cultural characteristics. And growth characteristics of Yersinia pestis may demonstrate some unique properties. It may grow on many growth media, even on the simplest meat peptone agar. But then the important thing is that it may grow, and you can see it here, in a wide range of pH that is not typical for bacteria. Usually bacteria grow within the neutral or slightly alkaline pH. And Yersinia pestis also may grow in a wide range of temperatures, from 2 to 45 degrees Celsius. Do you remember? In general microbiology, we studied that all bacteria can be classified according to the temperature requirements into three groups: psychrophilic, mesophilic, and thermophilic. So, Yersinia pestis can be recognized as psychrophilic and mesophilic also, but it is growing faster at 30 degrees Celsius. So then, several types of media are suitable for their primary isolation. For example, on McConkey's selective agar, the colonies are colorless because it is lactose non-fermenters. See, do you remember? The colonies of lactose fermenters look bright red on this medium, and the colonies of non-lactose fermenters look pale. So the colony of lactose fermenter most probably Escherichia coli, that is lactose positive. So Yersinia pestis is lactose negative. We also may cultivate on blood agar, and the colonies appear dark brown due to the absorption of hermine pigment, and they are very small after the growth at 24 hours at 37 degrees. So if the isolate is highly virulent, then it may produce viscous colonies due to the capsule production. Now let's take a look at the virulence factors. These are those factors I would like to remind you that allow bacteria to infect the organism, to stay in the organism and not removed from the organism. Then to multiply in the organism, to survive immune response, or to survive activity of the immune mechanisms, and then they also possibly spread in the tissues of the organism. So these are all stages of the bacterial infection. So when the virulence factors are those that allow bacteria to do this in the body. So for the first lipopolysaccharide, LPS, I would like to remind you that this is a component of all gram-negative bacterial cell wall. And from the point of view of medical microbiology, it is called endotoxin because it can cause very strong inflammatory response in the body directly by itself and indirectly through activation of macrophages. Then we have so-called fraction 1 substance, and this is the capsule envelope protein that is antiphagocytic factor, but it is heat labile because it is a protein, it has a protein nature. And I would like to draw your attention that these and other virulence factors most probably are encoded by not the major genetic element of bacteria, that is a chromosome, but usually by the plasmids. Because I would like to again remind you that plasmids quite many, the majority of plasmids are recognized as virulence factors because their products uh, of expression may help bacteria to cause diseases. So, fraction 1 is governed by the plasmid. Then, except for the antiphagocytic fraction 1 factor, we may have in the cell wall of Yersinia pestis V and W antigens that are again encoded by the particular PCAT plasmid, that is plasminogen activator gene. These antigens degrade the complement components, and you have to know that the complement is the protective mechanism of our innate immunity that involved in the localization, in the containment of the microorganism in the body and causing inflammatory response that is protective against these bacteria. And one more virulence factor is a coagulase. What is coagulase? This is the enzyme that causes blood or plasma to clot. And then this coagulase enzyme will be produced by Yersinia pestis at the temperature 28 degrees Celsius, but not at the 35 degrees Celsius. What is the sense of doing this? When the flea sucks blood from the vector, from the natural host, for example, rodents, mice, uh, rats, marmots, then the temperature in the gut of the flea is lower and usually in the hot or warm weather around 28 degrees Celsius. Bacteria start multiplying and producing the coagulase. And the blood that is gotten from the host uh, coagulate and block the ventriculus of the flea. So, and then so the food cannot pass uh, into the gut of the flea. So flea feels hungry and start biting everyone. So then one more virulence factor, fibrinolysin, that is also encoded by Pestis plasmid, and it degrades fibrin clot in the human or animal body and permit Yersinia pestis to spread readily in the body. Some more virulence factors. These are Yersinia outer membrane proteins that are incorporated into the outer membrane or the cell wall of this bacteria. And these are the components of type 3 secretion system. Do you remember we have discussed these secretion systems when we discussed Escherichia last year? And this is the effective molecular syringe that allows the bacterium to inject cytotoxic products directly into the cytoplasm of the target host cell. So the image shows a type 3 secretion needle of the surface of Yersinia pestis, when the product produced by bacteria will be injected through the membranes, bacterial membrane and human membrane, into the human cell. So directly. Then we also may have around 11 we have around 11 these outer membrane proteins. They are different. So, but mainly they code for cytotoxic proteins that may inhibit the migration and activity of the macrophages that's supposed to be phagocyte bacteria. And also they may inhibit the platelets aggregation that may help to stop spreading of the bacteria. So some more virulence factors we add, and this is pesticin, and this is the, um, we studied so-called bacterial sins. These are the substances that can be produced by one type of bacteria to inhibit the growth of other types of bacteria. So usually pesticins are not very toxic for humans, not toxic, but then they are toxic for the bacteria. What is the sense of producing this type of the organism? So Yersinia pestis may compete with some other microorganisms in the body, and then it may inhibit the growth to get some advantage to colonize the area for which it competes with some other bacteria. Pesticins is also encoded by the Yersinia pestis plasmids. Then Yersinia pestis keep on its surface the enzyme cascade, the components that gives Yersinia pestis the ability to absorb organic ion in the human body. And then finally, if we go to the top of the virulence factors, Yersinia pestis may produce so-called exotoxin. It is called murine toxin because it is highly pathogenic, lethal for mice, but is not so lethal for guinea pigs. And this factor is called fraction 2 antigen. And it is thermolabile protein and may be toxoided, so we can produce the non-toxic but still immunogenic portion of this toxin. So here you have summarizing slide where a lot of virulence factors are shown, and also the genetic mechanisms are mentioned. See, genes that code for these toxins or for these virulence factors. And also you can see what signaling pathways can be activated by communication of the bacterium with the human cell, especially with the macrophage. So then we go ahead of the clinical forms of plague. So usually we may distinguish three clinical forms of plague: bubonic, pneumonic, and septicemic. Out of these three, bubonic is the mildest form, and pneumonic and septicemic are highly lethal. Irrespectively over the site of entry, Yersinia pestis may then spread through the human body. Then it may cause septicemic plague. So it means that even bubonic plague that is transmitted through flea bite like this, see, entry through the flea bite, or if we have the pneumonic cases in the human population, the entry site can be the nasopharynx and pharynx because it can be inhaled by aerosol. Then, if irrespectively of the entry, it can be disseminated in the human body. So, when it may cause buboes formation, then pneumonia of internal organs, hemorrhage, and quite often the disseminated disease is lethal. So, few words about the most common disease, that is bubonic plague, most common clinical form. A flea usually feeds on infected rodents, and when they ingest bacteria multiply in the gut of the flea. And then bacterial coagulase block preventriculus of the flea. And this flea is considered to be blocked flea. It is called blocked flea because preventriculus is blocked and the blood cannot be sucked into the gut. So if this hungry blocked flea bites animal or it bites human, it regurgitate the blood during this, and by this the bacteria get into the body through the bite wound. So ulceration usually occurs in the in the biting site, and then we can see the wound as the set of the vesicles, then they start to be confluent and develop the ulcer. So then, from the site of entry, the inoculated bacterial cells are phagocytosed and multiply in the monocytes. So due to the virulence factor, monocytes cannot digest them properly. But then what they do usually, they carry them into the nearest lymph nodes. You can see here in this picture the uh, examination of the patient that might died from the bubonic plague. See, we have the multiple bulbous formation in the site of the lymph nodes. The inguinal lymph nodes are usually involved because the regular biting site for fleas are lower limbs. And the inflamed lymph nodes are called bulbous, meaning growing. So that gives the name for the type or form of this plague. Then hemorrhagic inflammation and necrosis may be developed in the lymph nodes. Also, they may rupture, and the pus will be released outside. But also, the dissemination through the bloodstream from the lymph nodes leads to the sepsis and the development of gangrene of the skin, fingers, and penis quite often, meningitis, pneumonia, pleurocarditis that may cause the death of the patient. A few words about the epidemiology of the plague. So in nature, it occurs in it was believed before 2 cycles, but now we know in 3 cycles. So we may distinguish sylvatic or wild cycle of the plague and urban cycle or domestic cycle of the plague. So in these two, the plague may occur independently of any humans, because this is in this cycle, for example, sylvatic cycle, the wild rodents occurs the natural source of Yersinia pestis, and then the bacteria will be transmitted through the population through the flea bite. So when human being enters this cycle or come closer in the environment populated by these infected rodents, then fleas start biting humans, so then humans can be infected. So the same in the urban or domestic cycle. So the domestic rodents like domestic mouse, they may live near the human settlements, and then the Yersinia pestis may circulate with the help of the flea bites. But then if quite often when the blocked flea appears, they have no and rodents die due to the enzootic or epizootic. So then the flea start biting humans, so then they can be infected. When the dissemination occurs in the body and bacteria infest the lungs, so then we have the cases of pneumonic plague in the body. Then if we have the cases of pneumonic plague, the disease by spreading Yersinia pestis can spread directly from human to human without any fleas. So then it may be considered as a separate cycle of the plague. So because it can be transmitted since the appearance of the pneumonic cases of the plague directly from human to human. Few words about the laboratory diagnosis of the plague. As we discussed, it can be seen by microscopy, and usually we may perform staining with methylene blue or Giemsa staining. This is Giemsa staining. This is Giemsa staining, or it can be stained by immunofluorescent technique, so we can see it under immunofluorescent microscope. But then this will be not enough, so it should be cultured. That is the golden standard for diagnostic techniques. They're supposed to be cultured, and all cultures must be handled with the extreme caution because, as you heard before, it belongs to the fourth biosafety level with the highest risk of contamination and dangers. So the materials can be cultured also on McConaughey's agar and blood agar. Serological tests can be used, and this is passive hemagglutination test and complement fixation test to identify the antibodies against Yersinia pestis. The detecting of the antigen in the human tissues can be performed also with the complement fixation test and immunofluorescent techniques, but also by some precipitation test that we will study in immunology course. So usually, if you would like to uh, be sure that this is the Yersinia pestis microorganism, we do animal inoculation using guinea pigs to demonstrate the high pathogenicity of the microbe. So we have discussed already that the plague can be treated with the old good drugs like streptomycin, tetracycline, gentamicin, and some others. And the development of drug resistance in Yersinia pestis is rare because nobody really treats the wild animals with antibiotics. So, and the prevention of plague is also possible. So, for specific prevention, we use vaccination, and formalin treated whole killed vaccine is available. So, and it can be used as pre-exposure prophylaxis. So, usually we selectively vaccinate those people who handle animals, laboratory workers, or travelers to endemic areas, and this is called pre-exposure vaccination, but not really post-exposure vaccination. Live plague vaccine is also available. This is a Russian STI vaccine that contains the vaccinal strain, and usually single subcutaneous administration occurs. But the thing is that it doesn't give 100% protection. So the whole killed and live plague vaccines uh, will protect from bubonic plague, and they will be effective for from half a year to 1 year, and less effective or non-effective at all against the uh, pulmonary plague. So from Yersinia pestis, we go to Vibrio cholera. That is a causative agent of cholera. So because we give just an overview of those bacteria and some details you may later on take a look at some details using any microbiology thick medical microbiology thick book or department book. So cholera is a potentially epidemic and life-threatening secretory diarrhea characterized by numerous voluminous watery stool, often accompanied by vomiting and resulting in hypovolemic shock and acidosis. And that is the final result of the severe cholera. Let's take a look at the Vibrio itself. It is quite small, gram-negative, curve-shaped bacilli with one single polar flagellum, and due to the presence of this flagellum, they are highly motile. According to the oxygen requirements, they are facultative anaerobic bacteria. But Vibrio cholera itself quite often is obligate aerobic. But the related species from Vibrio are facultative anaerobic. So again, Vibrio cholera, that's also typical for this. It may grow within a broad range of temperatures from 18 to 37, not that broad like Yersinia pestis, but still. And this temperature can be used for selective isolation of the organism from species contaminated with some other bacteria. For example, when we try to isolate it from water, because it lives in water. Vibrio cholera is susceptible to heat, drying, and acidity of the gastric juice, so it cannot be easily passed uh, through the gut. It also may survive for months in sterile seawater and for months in a tap water. And it is interesting that Vibrio cholera may easily enter the state that is called non-culturable state. And that's funny that Vibrio cholera may occur in the in some liquid, for example, in the distilled water. But if we put it in culture, we won't see any growth because it enters their non-culturable state. Few words about the Vibrio cholera classification that is really very complicated. So, for the first, we classify Vibrio cholera. So, my dears, I would like to underline that now we are talking about species Vibrio cholera. But truly, some other species of Vibria occurs. For example, Vibria parahemolyticus, or Vibrio mechnikovi, or Vibrio some others. My dears, these are very related to Vibrio cholera bacteria that are collected in one single genus, Vibrio, that is a genus. But then out of all species in this genus, Vibrio cholera is most dangerous and pathogenic and can be recognized uh, potential warfare agents, not the others. Okay, so that's why we are talking about Vibrio cholera itself. So then Vibrio cholera that is considered pathogenic. Do you remember that the pathogenicity of bacteria is identified on the species level, but different isolates of Vibrio cholera or different cultures of Vibrio cholera isolated from different sources, or we call them strains, can be of different virulence, more virulent or less virulent. That's, I remind you, the infection and infectious process topic from the general microbiology. Okay, now let's take a look at the classification. Based on the antigenic structure of the cell wall of Vibrio cholera that are gram-negative, so they have lipopolysaccharide as a part of outer membrane and somatic antigen on the top. So based on the antigenic structure of somatic antigen, this is OR, we may distinguish 130, even more than 139 serogroups. Okay? So serogroups, see, from O1 to or even 141. Some of them can be associated with some outbreaks, but we distinguish two highly pathogenic serogroups, O1 and O139. These are of utmost importance. All others in between, from O2 to O138, are not highly pathogenic. They may cause some sporadic diseases, but without very severe symptoms. So clinicians don't care about them really. That's interesting for researchers only. So then when we are talking about pathogenic microbes, the assumption is that they belong to O1 serogroup or O139 serogroup. So then, these two and the rest are called non-agglutinable Vibrio cholera vibrios, right? Non-agglutinable because they won't give positive reaction, immunological diagnostic reaction with antibodies against O1 somatic antigen or with antibodies against O139. We'll discuss some diagnostic tests within the classes and within the immunology classes. So, okay, these two Vibrio cholera serogroups are highly pathogenic, capable of causing cholera. The rest cannot cause cholera itself, the disease that is called cholera. Out of these two, O139 is very rare now, it is hidden somewhere in nature. So O1, this is the one that may cause outbreaks from time to time in different sites of the regions of the world. So then O1 in turn, the only this serogroup can be further subclassified into serotypes. And we call them Ogawa, Inaba, and Kojima. And these are serotypes that usually cause cholera. Okay, this is one mode of classification based on the antigenic structure. The other way of classification, and this is about O1 only, serogroup only. See, Vibrio cholera O1 now. So it can be classified based on biochemical tests and susceptibility to bacteriophages. And traditionally, Vibrio cholera O1 is classified into two so-called biotypes of biovars. They are called classic or classical and Lektor biovars. And even Lektor can be further classified. We won't discuss it. It's too complex for you, okay? So then you have to know two biotypes, classical and Lektor. Classical is the old one. Now it is hidden somewhere in nature, it disappears. It used to cause pandemics before, but now it is hidden somewhere in nature, and Lektor biovar now is a causative agent of cholera around the world. So this is a comparatively new biovar. that replace somehow the classical biomarker. So, but then we always need to confirm whether the cholera disease is called by Hector and or classical. Maybe it returns once. Okay, then few words about the pathogenesis of the disease. So usually the way of transmission is fecal oral. And the typical way is the ingestion of large number of microorganisms with contaminated water, because usually it lives in, for example, Ganga River, in fresh waters, or in river estuary with a very low salinity. So, and also food contaminated with the with fresh water, contaminated water may serve as cause of infection. The thing is that gastric acidity provides a quite effective barrier against infection. So, but for a person with achlorhydria or hypochlorhydria, the infective dose can be very small. And it can be reduced from 10 power 3 to 10 power 5 organism. So it means the people that have some predisposition for the infection, the infectious dose can be lower than for those who have normal acidity of the gut. So in the small intestinal tract, Vibrio crosses the protective layer and reach epithelial cells. With the help of high motility, this is a virulence factor of Vibrio cholera. Then chemotaxis, adhesive villi that allow to adhere to the mucous membranes. Then enzyme mucinase that make uh, easier for bacteria to reach their target cells. Then what happens in the small intestinal tract? Cholera start producing the highly potent toxin that is called cholera gem. And you can see here this toxin is of AB type, and we have 2 A subunit and 5 B subunits. And I would like to remind you that A subunit is called so because it is active portion of the toxin, and B subunit is called so because it is binding portion. So you can see the microbe that start producing toxin. This is a toxin that by B subunit binds with the special ganglioside receptors on the surface of the human cell. And then the through binding with the GM1 receptor, the toxic molecule can be internalized by endocytosis into the cell. And then it will be transported to Golgi apparatus. And due to the B subunits, the fusion occur with Golgi apparatus, and the toxin will be emptied from endosome into the Golgi apparatus. Then there, the subunit interacts with G protein, and G protein target activation of adenylyl cyclase, and it converts ATP to cyclic AMP, and it causes the hypersecretion of water electrolytes from the cell into the lumen of the gut. And that is the cause of very severe watery or so-called dry stool diarrhea. And this diarrhea can be so so severe that the person may may lose around 10 liters of water per day. And you have to understand that the person with this diarrhea start to be so weak very quickly that this person cannot even drink itself. That's why it is very important to help the infected person to perform rehydration therapy. And that is of utmost importance, even more important than antibiotic treatment, because the loss of water is a factor that causes really all symptoms of cholera disease. So that's why it's so important to perform rehydration therapy. I would like to strengthen it. And the rehydration therapy should be performed with water supplied with electrolytes. So the electrolyte balance should be uh, renewed or maintained. So, how do we perform the diagnosis of the cholera disease? My dears, truly, the diagnostic technique is very complicated. It depends on where we want to isolate cholera, Vibrio cholera from. Whether we want to isolate it from water, because by the way, in areas where cholera is endemic, for example, Ganga River. So the special sanitary stations have to monitor the presence of this Vibrio in waters. So it means they every week or every second day or whatever, more or less often, they need to try to isolate Vibrio cholera from waters. So that's why it is performed very often. Or, for example, another sample can be the feces from the patients. So that's supposed to be diagnosed for cholera. So that's why the stages of diagnosis depend very much on what sample is taken for examination. But without any doubts, the culture is supposed to be performed. So this is a still golden standard for cholera diagnosis. As soon as culture is performed, the cholera, Vibrio cholera, should be classified or identified. The antigenic structures should be identified by agglutination tests, and the classification into serogroups supposed to be performed. And then biochemical tests are also performed to identify Vibrio cholera and to discriminate it from any other Vibrios that also can be found in the waters. Then also when we get culture, the biotyping should occur into whether it is classical or L-Tor biovar. And also phage typing may help in classification into biotypes. Apart from traditional culture technique, the polymerase chain reaction is used now for express diagnosis, for very fast, rapid diagnostic techniques. And interestingly, the PCR tests are developed to identify several targets or several genes in the microorganism taken directly from the environment or directly from the clinical specimen. And usually with the help of PCR, we would like to identify the genes that code for O1 antigen of lipopolysaccharide. There are also genes uh, that are important for adhesion of these microorganisms to the human cells. and the gene that code for cholera toxin. Usually we perform triple PCR. In the one single uh, test, we identify three different genes. If all three are positive, so we may get the positive result for cholera diagnosis very quickly within the one working day. And of course, it is much faster and much better than a culture technique within at least 3 days. So this is a great advantage of the PCR tests. So both types of tests are available right now. PCR technique and culture of isolation of bacteria from the specimen. With this, I would like to finish the diagnostic procedure and tell few words about the treatment and prevention of this disease. So we stated that if not treated, uh, cholera as a disease can be life-threatening. But it is easily treated by immediate rehydration, that is replacement of the fluid and electrolytes, and antibiotic treatment that shorten the duration of the illness and lessen the severity of the disease. Several injectable vaccines are available that are used for specific prophylaxis or prevention of this disease. And they can be recommended for those who live in the endemic area or for travelers to high-risk countries. So this vaccination may protect against cholera within several months, but not for very long while. So that's why this vaccine is not in the very not really often used for the prevention of Vibrio cholera disease. Now we come up to the third microorganism that's supposed to discuss today. That is Bacillus anthracis and the disease that it causes that is called anthrax. On the very first slide regarding this infection, you can see the microbiome lab where Robert Koch had been working when he discovered Bacillus anthracis and proved this is a causative agent of the disease. And you also can see microscopic examination of the sample, and you can see the long rod-like bacteria arranged in pairs or chains. That is typical appearance of these bacteria. So, Bacillus anthracis causes anthrax, and this is an important disease of animals. And from time to time, it may also occur in humans. So, and that's why it is called zoonotic disease. And truly, the Bacillus anthracis itself is recognized as a major bacterial agent of biologic warfare. So I think they consider the major reason because it may produce spores, and the spores are very resistant in the environment that cannot be easily eliminated from the environment. That's make Bacillus anthracis very possible organism that can be used independently as warfare agents. Let's take a look at the Bacillus itself. The genus Bacillus is highly populated with large aerobic gram-positive rods. And all out of this genus, all bacteria have square ends, as you see here, and are arranged in short or long chains. And they all may produce spores that can be located centrally terminally or subterminally, centrally very common. Very common for bacillus anthracis. So usually these bacilli are generally motile, but Bacillus anthracis itself is non-motile. So in this genus, we may find psychrophilic, mesophilic, and thermophilic species. So in there we have quite a lot of saprophytic bacteria that utilize simple sources of nitrogen and carbon for energy and growth. But not all of them are of medical importance. So, out of saprophytic bacteria, we may consider Bacillus subtilis and sometimes cereus. But we will talk about Bacillus anthracis only because it is the only principal pathogenic species of this genus Bacillus. It was first Bacillus to be isolated in pure culture and shown to process spores by Robert Koch, 1876. And it was the first bacterium used for preparation of attenuated vaccine by Louis Pasteur, and it was done in 1881. See, these two Nobel guys both worked with Bacillus anthracis. And this is morphology of this bacilli. So, few words more about the morphology because Bacillus anthracis may produce several types of structures. So, usually when it resides in human body or animal body, in tissues, in live body, they may produce capsules, my dears. The capsules are not produced in dead bodies and are not produced in soil, for example, right? So, in living bodies only. So, and usually the vegetative cells come together by square ends and it is believed that the appearance looks like bamboo sticks. So the capsule is made of polypeptide and that's why it is thermolabile. Uh, when blood films containing anthrax bacilli are stained with polychrome methylene blue and examined microscopically, we can see the purplish capsule material around the bacilli. But this is called McFadden reaction now. It is not widely used in the diagnosis when we perform it in human, mainly in animals. But Bacillus anthracis capsule can be seen by negative staining also and by immunofluorescent staining. You can see how it appears. If we are dealing with the Bacillus anthracis encapsulated organism, then when we grow it on the surface of the MPA, for example, they give very specific uh, mucoid colonies. You can see, and they even confluence sometimes when we take a look at it on the surface of MPA. But usually, in all ordinary conditions, when we perform culture of Bacillus anthracis, the capsule is not produced, is not formed by these bacteria because it's nothing to be protected from in the culture. But we may force these bacteria to produce capsule. Why it is important? Because when we perform diagnosis, we have to confirm that Bacillus anthracis that we isolate can produce capsule. So to make it visible, to force the microbe to produce capsule, we may develop special media. We may add bicarbonate into the medium and cultivate in the under conditions with high percent of carbon dioxide, or we may add serum, albumin, or starch. Then bacteria start producing capsule. So then another structure that is produced by Bacillus anthracis is a spore. You can see the spores here. They are elliptical and located centrally. And the single spore, when the rest of the vegetative cell is autolyzed. And you can see that the spores are the same width as bacterial cell. I think you know already what is a spore. Sorry. Bacteria can produce one single spore, and this is produced by condensation of the layers around the chromosome. And the endospore is highly resistant in the environment. Why? Because it is covered by several layers, and you can see the name of these layers here. And one more thing, it has no metabolic activity. That's why it is so resistant in the environment. And the spores will be produced in the dead bodies within several hours after the death of the host. So because the conditions start to be not so good, and the spores are formed usually in non-favorable conditions. So when one is dealing with the carcasses of animals, then the spores will be there. Or with the soil contaminated with Bacillus anthracis, then the spores will be there. A few words about the cultural characteristics. This organism is aerobic and uh, it may be cultured in a range of temperature from 12 to 45, and the optimal is 35 to 37. It may grow, it may give deposit growth when it is cultured in the liquid medium. So because when bacilli will be arranged in these bamboo sticks, those structures are very heavy, so they fall down the bottom of the tube. So the liquid can show no turbidity, the culture medium, but the deposit will be seen on the bottom of the tube. So Bacillus anthracis, this is an organism that can be grown on in ordinary medium. When it is grown on the uh, petri dish on the hard agar, it gives the appearance of cut glass in the transmitted light. And why it is important? Because when we perform the culture of Bacillus anthracis, we have to recognize that this is Bacillus anthracis as soon as possible. So to keep the safety precautions in growing this. Then Bacillus anthracis may form S and R colonies, and the R colonies are rough, grayish, white, and the age of the colonies um, mm, are composed of the long interlacing chains of bacilli. You see, these are bacilli arranged in chains. So, and the appearance of this type of colony is called medusa head appearance, and we consider medusa horn. Do you remember the creature, uh, mythological creature with the hairs from this made up of snakes? That's medusa head appearance of the colony of Bacillus anthracis. So when we culture it, when we grow it on the blood agar, Bacillus anthracis doesn't give any hemolysis. So, and this is the differential feature to differentiate with Bacillus cereus or some other bacilli that can be hemolytic. So Bacillus anthracis also may produce the proteolytic properties that are not very common for bacteria generally. When we inoculate the gelatin column with the Bacillus anthracis, after several days, we can see the liquefaction over the gelatin that gives the appearance of the inverted fir tree, see, or ivy tree, like this. So, gelatin liquefaction is the test that is used to demonstrate the proteolytic properties of bacteria. Then also for for Bacillus anthracis identification, we use so-called string of pearl reaction. Because normally, when bacteria are grown on medium without antibiotics, without penicillin, uh, it will give normal morphology of bacteria. But if we subculture it on the medium with penicillin, to which it is highly sensitive, after 3 to 6 hours, cells become larger, spherical, and occur still in chains, so it gives the appearance of the string of pearl necklace or bracelet. So, but then if again we subculture this bacteria on the medium without penicillin, they retain the original morphology of bacteria. So this diagnostic reaction. Usually, if we want to perform diagnosis, we have to culture these bacteria. But quite often we need to use selective medium. So we have a special so-called PLET medium that contains polymycin, lysozyme, EDTA, and thallus acetate with added heat, hot infusion agar. So, and that is a very special medium that allow to discriminate Bacillus anthracis from a mixture containing other spores bearing bacilli. The biochemical reactions that can be sometimes useful for isolation of the identification of Vibrio cholera are sugar fermentation tests where that can be performed, fermentation can be performed with acid but no gas. But high carbohydrate fermentation is not really very useful. Bacillus anthracis highly resistant in the environment. And the experiment had been done for keeping Bacillus anthracis spores in the soil for about 6 decades, and they were still alive. They withstand dry heat and certain chemical disinfectants for a moderate period, for several weeks, for example. That's because the spores. If you want to perform sterilization of the bacillus containing materials, so the animal products and other materials should be autoclaved because most heat under high pressure is the best way to kill bacterial spores. So a few words about the anthrax as a disease. It is a disease of herbivores. Other animals are relatively resistant to this infection. And humans usually can be infected incidentally by contact with infected animals or their products. This is one of the serious biological agents considered as warfare agent. The sources of infection are those animals in nature. You can see them, right? Quite a lot. So, and there are several virulence factors of Bacillus anthracis. We mentioned some already. The capsule that is antiphagocytic. One of the most important is anthrax toxin that is called exotoxin, and it is made up of three proteins: PA, protective antigen; EF, this is the edema factor; and LF, this is lethal factor, that is major virulence factor, which caused the death of the animals. That's how the toxin works, the mode of anthrax toxin action. You can see when Bacillus anthracis multiply, they produce these subunits of the toxin. The protective antigen, this is a subunit that binds with the anthrax receptor, and then the polymerization of these subunits occur, you can see it here, and then it make a platform for binding of lethal factor and edema factor. Then all those structures will be engulfed by endocytosis, and then from the endosome, with the help of protective antigen, the edema factor and lethal factor will be released outside into the cytosol of the cell. Edema factor causes the activation of cyclic AMP and the release of fluid in the interspecies space that may cause edema. And the lethal factor is the metalloprotease that causes necrosis and hypoxia of the cell. So pathogenesis of anthrax. There are three ways or three clinical forms of anthrax. Cutaneous anthrax, that is most common. Uh, when the spores contaminate the injured skin, and we have low-level germination and growth of bacteria, uh, first spore, then bacteria in one side, but then it may also cause for local edema and necrosis. But then it may cause the infection of macrophages and spreading of bacteria in the body. Then bacteria may enter through intestinal tract. In humans, it is very uncommon, very rare. So there we have also low-level germination at one side that results in massive effusion, mucosal edema, and necrotic lesions and infection of the macrophages that come up to the area to engulf bacteria. And the third way is a pulmonary inhalation of the spores. And also it may lead also to the contamination of macrophages, where the virulence factors will be released like capsule, exotoxins, and other factors. Macrophages will take bacteria to the regional lymph nodes. Then there we have regional hemorrhagic lymphadenitis, then septicemia, toxemia, and then the very severe uh, mm, results of the Bacillus anthracis infection. So pulmonary lymphatic blockage, edema, and death. So septicemia, toxemia, shock and death, or meningitis and death. So why I would like to tell few words about why the disease is called anthrax. Anthracite is the charcoal that is used to to burn and warm the environment, right? And it is a stone with shiny black appearance. So if we have a cutaneous anthrax, the wound that is formed in this area resembles charcoal. That's why this disease had been called anthrax. So cutaneous anthrax is the most common and acquired through injured skin. Inhalation anthrax is required through inhalation of spores into the lungs. And gastrointestinal anthrax, the entry of spores through the GIT. So here you can see the appearance of the anthrax carbuncle, and the wound results from the Bacillus anthracis infection is characterized by the edema around the wound and the necrotic appearance of the scar of the wound. So we have discussed the pathogenesis already, and this necrotic ulceration, black ash scar that appears as charcoal. It may heal, but it takes very long while. So the approximately 5% of cases in humans are for inhalation anthrax, and the rest are for uh, cutaneous anthrax. Gastrointestinal is very rare. The inhalation anthrax is very severe disease. So with very long incubation period, but very fast appearance of the clinical symptoms, and quite usually it is fatal. So my dears, I feel like we need to finish our lecture because time is up. So we discuss almost all features and take a look at the diagnosis in a microbiome book. With this, I would like to finish my lecture and see you in classes. Goodbye, my dears.